Yeah, I think we just thought it would be useful for the kind of concrete implementation of behavior in Monty, kind of really nailing down how it should be represented and what it really means and stuff. And we thought that could touch a little bit on both time cells, sorry, matrix cells and time cells. Um, but yeah, Scott, do you want to take
Yeah,
over?
that's a pretty good summary. Or, last week, basically, Vivian laid out a 4-point plan. A couple of them actionable. Uh, I say as actionable as in like a couple of them concrete things we want to be doing shortly with regards to object models, learning them and recognizing them, and then laid out some ideas for how to build associative connections between behavioral and morphological models, and then Nils also gave presented a discussion about motion and time, things like that, and we thought we would sort of let Vivian take the floor again, and we can continue the discussion about behavioral models and time in particular. So let's go ahead and uh, take it away. I yield the floor to my colleague from Greece.
Oh, that's too big of a build. I unfortunately didn't have as much time to prepare as I would have liked to have. Um, so yeah, I started making some slides, but actually I might just go through the papers. But yeah, basically after last week's meeting, I wrote up this document that I think most of you have already looked at, but basically outlining those four capabilities and then what we would need to do concretely in Monty to get them. And I feel pretty confident about the first three capabilities and being able to implement that in Monty. But there's one kind of bigger question mark around them, which is all about time and how that should be represented in Monty and how that kind of clock signal should look like and uh, out from outside into the learning module and how the learning module internally should represent time. And yeah, there were a couple of longer messages between Jeff and me, and I think there's, at least in my mind, still a couple of open questions around that. So yeah, that's kind of the main topic I would like to talk a bit more about today, because if I do have more concrete answers, then I feel like we could start doing a first prototype of modeling object behaviors. And I guess in the scope of that, I had a bit of a look into the literature on matrix neurons or matrix cells in the thalamus and their properties and kind of what's known about them. And I started looking into generally time processing in the brain and different mechanisms and places that can be happening, but didn't really get to complete that part. So feel free to just jump in if you have other things you want to talk about or bring up, but I would just kind of go over the literature briefly and we can go on any of these figures in more detail if you think it would be useful. Maybe they bring up any ideas or thoughts of how it could fit into the theory.
Yeah,
And we can just
that
go
sounds good.
quickly over it.
Yeah, and maybe one thing that kind of came up at the research meeting on Monday, which may not be right, but just a potential framing that we were discussing then, Jeff, is that the matrix cells are kind of the sort of what's bringing us through a sequence and sort of kind of telling us the time is passing basically, and that the sort of time cells are a bit more like place cells. They're a bit more kind of like where you are in, like what is your actual behavioral state and kind of tying you to a point in that just based on, but yeah, we can revisit some of that
I
later.
know
But
almost nothing about time cells, so
okay, yeah,
in
I
fact I
listened
do
to a
know nothing
podcast
about
about
them.
them and and read a little bit about them at one point, and I seem to remember they were a little like place cells. That it was kind of like, there's kind of a almost like a Gaussian or kind of like a lopsided area, but in time that they kind of respond to.
Yeah,
Um,
before we get, I mean, I don't want to go off track here, but one of the things I think about time is that does it have an, is there like a start to time, like, time from when?
Yeah.
It's a little bit like grid cells in general. Like grid cells, we always think as engineers or whatever, we think of like, oh, there's an origin point. And it's not, that's problematic in so many ways. And nature doesn't seem to have that, right? It figured out how to do locations without some sort of origin. And I always felt time is the same issue. It's like you don't really want to start time from some origin, but it should all be relative.
Yeah.
So
So
I
it
don't,
kind of needs almost like an anchoring point.
well, my point is, is there an anchoring point? Is there some clock that begins when you begin a sequence or a melody and it can play throughout? I've always thought that would be problematic. So again, I don't know anything
But
about
don't we
time
have that
cells.
with space, both with like place cells and grid cells? They kind of re-anchor relative to something in the world.
Right, but there's no origin, right? It's the origin in time would be time zero. So they could be anchored like in the way that you could I suppose, I don't know, again, I know nothing about time cells. So I suppose you could have cells that are unique time, some time and some thing at, I don't know, that's unique to an object or unique to a sequence, or it could be not unique. I just think when I started thinking about melodies, what I realized was you don't having like a clock from the beginning doesn't really make any sense. What you really need is an interval clock, and from point to point, and you need to be able to modify all of them. So I should just stop talking here because all I'm pointing out is like I think if I think about, okay, what's modeling have to do with time, I would talk from a very high level perspective. Okay, what is our philosophy of time? What are we timing from? Is it the beginning of a sequence and we're marking points along the way, or is it like point to point to point type of thing?
Yeah,
But maybe,
my guess, and maybe we can get more into this discussion later, or like it feels like it would definitely be relative, but whether it's relative to could constantly be changing. Again, a bit like kind of re-anchoring, so that
right.
it's not just like the beginning of the song. The beginning of the song is maybe one thing you can anchor to for a bit, but then maybe that changes what you're kind of
When the
anchored to.
solution I came up for melodies was like, oh, there is no anchoring time in the beginning. You have no absolute sense of where you are time wise. All you have is
But don't you know that you're kind of at the start of the song?
well, you can know when you're at the start, but when you're in the middle of it, you have no sense of how far into it in terms of you can always go back and recreate it. You can start from the beginning again, but
You have a bit of a
it's
sense
like
though.
saying,
Like,
oh, I say, well, let's start at 3 seconds into this or 30 seconds into this song. You have no idea where that is. You just know it's not the beginning at that point. So I don't think we really have a sense of this clock running through time. We have, and so I'm just curious what time cells actually represent. What are they?
Yeah,
Yeah.
actually, Scott made some slides about time cells a couple months ago, but never got the chance to present them. So maybe this might also be a good chance to talk about some of the research on those
Yeah.
today.
I have those slides pulled up. I could go over any of that sort of stuff if people want me to jump in for a minute. But the basic idea is like the findings of the time cells in the hippocampus is sort of like there would be a start point, a re-anchoring point at the beginning of some kind of stimulus, significant stimulus or event or contextually important event. So the beginning of a experimental trial in which there's a big beep or flash of sound or something does seem to start off some sort of cascade of time cells. So I think
That's consistent, by the way, with my vision of the matrix cells, right? In a melody, you basically every note is an event, or every beat, every significant note, and then you have a clock that plays out, and then you start again, a clock that plays out. I don't know, maybe time cells go longer periods.
well, my understanding is, so if we say matrix cells, if this is even right, are kind of more like a metronome, kind of like a beat. Time
I don't think
cells
that's right.
are okay.
That's not how I've always tried to get. That's not how I view them at all.
But like, sorry, a metronome that kicks in from like a starting event.
It's a shot clock. It's a countdown from 1 second, or a count up from
Yeah.
1 second. It's not a metronome. It doesn't have any kind of regular pattern to it. It would be, look, first of all, when we talk about matrix cells, we state once again, this is a pure hypothesis, unproven. Okay, but it fits what we know about the physiology of melodies. So it would be more like it's not in the background doing anything, but when you have an event, it sets up a cascade of activity over a population of cells. That cascade dies down after about a second, and then it waits for the next event. So that's the hypothesis there. So there's no metronome going click, 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 click. It's like, oh, an event, countdown. All right, here's another event, countdown, type of thing.
Okay, that sounds, yeah, more similar to my understanding of time cells, but maybe you can show those slides, Scott. But my understanding of the time cells is like you basically have different ones. Again, if you imagine a place cell has kind of like a kind of Gaussian in space of where it's sensitive. Time cells, it's a bit more like, oh, I'm a time cell that's sensitive to like a few seconds after something happens, or I'm a time cell that's sensitive to a few minutes after something happens.
Do time cells go out that far?
I think
Well,
they
they've
do. I
thought
think
to
it's like
go out a
logarithmic.
ways, but they become less and less sort of accurate as time goes on.
How
So,
far
like,
out? How far out do they go?
I'm not sure if it's been measured exactly. Well, I'm sure some people have tried, but I would say on the order of seconds.
Yeah.
So this actually goes out to the top 16 seconds here, actually. But it gets messier and messier as the further you go out. So shorter durations are more densely represented and more.
Right. Well, this is consistent with the mechanism that I was imagining for matrix cells, except the evidence we have from melodies, you can't time anything more than a second. So, but it's basically the same mechanism, an event followed by a series of cells that are activating and sort of Gaussians, and the further out you get, the less accurate it is. So, I could easily imagine that if these are in the hippocampus, the same basic mechanism is used, but in the hippocampus, maybe it goes for, 20 seconds. I don't know. That didn't seem to be the case, at least, from the observations of melodies, people can't do that. So, but it could be the same basic neural mechanism, just of different timescales.
Yeah, I think it's hard to know what the longest durations possible are. I'm sure it just gets harder and harder to measure the longer durations in the hippocampus. But they've kind of looked at it and entorhinal cortex and found similar kinds of things, and the findings in the neocortex are fuzzier and harder to
So on
find.
this little chart here, it says normalized time. So that doesn't tell us anything then, does it?
I don't remember the details of the study. I don't know
Well,
what
it's normalized.
so who knows what that,
it goes to
I don't
1.
know what the length of the trial was supposed to be. So yeah, that could be
I was thinking,
long,
oh, look, it goes 1 second. It goes, oh no, that's normal.
Yeah. Yeah, hippocampus is, as far as I'm aware, it's really the place where the findings are like ironclad, and we're looking more and more in other parts of the cortex, but that's sort of a newer area where things are less clear. There's been less convergence in the field about the results there.
One high level thought is, just like we kind of realized at some point in time in the past that every part of the cortex is dealing with locations in space and reference frames. And then it seems like, oh yeah, of course, it has to be like that. It might be the same thing with time. We might be thinking like, if you have movement and you have, changing patterns in the world, time may be just an integral part of everything. And so we would expect to see, time-like cells, throughout the brain. The difference with well, at least when I'm thinking about melodies, I realize that it has to be centralized for certain areas because you want to speed up and slow down all the cortical columns, all the learning modules that are processing the same input. Anyway, maybe I'm just saying we shouldn't be surprised if we find time-like cells everywhere.
Yeah, that's kind of what I found in the literature too, that there's like representations of time pretty much everywhere, and that the brain uses several different types of mechanisms for like different time scales and different purposes, like exact timing of motor outputs or learning temporal structure of sensory inputs.
Well, I always
Yeah.
imagined that the same timing mechanism used for melodies would be used for fine motor control. Right. When you're exercising something like, you're signing your name or you're doing any kind of task, some parts, your body moves at different rates at different points. There's a timing to the sequence, and you can generally speed it all up and you can slow it all down. Like I could speak very rapidly or I could speak very slowly, but the relative timing of elements would be pretty much consistent. And so that struck me, okay, it's probably the same mechanism that we use for inference, like melodies we would use for motor output. Same basic requirements.
Right. And then I guess in terms of kind of how the information is encoded and communicated, do you imagine, Jeff, it looking a little more like this? That, I appreciate there's limitations to just measuring individual cells, but like here, they find this pattern of like, oh, okay, some cells respond quite like early. They've kind of like specialized for early after an event, and some cells are specialized for kind of later after an event.
Right.
Is that
right.
kind
well,
of the substrate, or is it because I think maybe that's, I don't know if you're the same, Vivian, but at least my mind was going down the kind of metronome or just because like when I think of a shot clock, I think of like, oh, okay, now 1 second has passed, now 2 seconds has passed. Like
maybe I shouldn't have
and
used the word shot clock. Maybe that's the wrong thing.
yeah,
Maybe I made that up. I thought that it was more like a 1 second time down thing. This is exactly how I imagined it. Look, I had no evidence for this, none whatsoever. I had no knowledge about what matrix cells firing patterns look like. I chose them only because they fit the physical connection role, and then no one else had any idea what they did. So I said, okay, good guess. But then I'm saying, okay, well, what you really want, imagine I'm a neuron and I'm trying to detect when a certain amount of time has passed. And I have to learn that. I have to form synapses to that time. So it's just a bunch of axons. So I always imagined that you'd have a set of axons which become active in sequence. Not like one, then the next one, then the next one, because neurons don't work like that. You'd always have to have coactive ones set. So I imagine just like this. I imagine you'd have a set of axons. These are the matrix cell axons, again, making that up. And that in the beginning, some would be firing and then they would stop and some others would fire. And so there'd be just like you see here. This is exactly what I imagined it would be. I didn't anticipate that you might have the spreading of time as you further got out. Like it could have been more of just a linear sequence as opposed to getting wider and wider their responses. But that's not inconsistent.
That kind of makes sense, I guess, in terms of precision becomes harder the longer the scale.
Right.
Like it
so
seems
maybe
kind of, yeah.
It gives you a little bit more flexibility. It means that the longer you wait, the easier it is to catch the right time or something like that. I don't know. I didn't think about that much. But this is exactly very, very close to how I imagine matrix cells could work.
And then when you speed it up, would you think of it still being, because I imagine when you learn the sequence, you would learn specific associations of like the next event happened when this neuron fired.
Well, a bunch of them, right? A set of them.
Yeah, but if you speed up the sequence, then a totally different set of neurons would be firing after that time.
No, that's not what I imagined at all. It's the same set of neurons. They just get compressed.
I think Jeff is right about that in terms of the literature. Like I have to go back to look at this 2013 one again, but I'm pretty sure when they actually shortened the trials, like the delays between like go and act, this line here would just like the neurons themselves don't have a fixed time constant, like a fixed delay afterwards.
Okay,
like they would actually adjust and
right. So that red line would get more vertical.
Exactly.
Okay, perfect. Yeah, that's exactly what I was hoping to be possible. Cause so basically the x-axis isn't really time in seconds that they encode. It's more like position in the overall interval
Well,
I
it
can
is,
represent.
it is time in seconds unless you modify it, right? I mean,
Yeah, but
in
you
the
can
case of the
modify.
melodies, there is a normal, natural time sequence. And that's why we know exactly where I hear a melody. I know the tempo and the timing exactly of how I heard it. It's not lost to me, but I'm able to speed it up or slow it down as necessary. So there's like, you can imagine there's a standard clock cycle like you're showing these pictures here, but under certain circumstances, if I have to go quicker, I can speed it up, or if I'm listening to something and it seems to be slower, I can stretch it out. But there is a base timing sequence, and which can be sped up or slowed down. That's the evidence from melodies.
And
Nice.
so
Well,
when
yeah,
I
good.
see, like, if a column basically wants to speed up the melody, it knows, oh, I sent the signal to speed it up, so I know it's being played at a faster speed now. But what about the other columns that receive the same kind of global signal?
Well, it of course it doesn't know anything, right? Columns can't think, but but that's why the whole thing is, it's centrally located, right? So anybody can say, hey, you know, my note was my note came too early, a little bit too early, you know, and therefore that's sent back to, let's say, the matrix cells, and the matrix cells just speed up their time constant a little bit, and then it's sped up for everybody. So I don't think there's any local knowledge in the learning module or in a column about the tempo. There's no local knowledge about it. It's just like, oh, let's say I have a rhythm. I know that these notes are, you know, half a second apart, and but at any point in time, anybody could speed it up or slow it down, and if that fits, it fits. You know, there's no, I don't have an internal knowledge about how fast things are going.
So the fact that we know the kind of default speed of a song is just, that would just be because there's some kind of default speed of the matrix cells
Imagine inside the matrix, in the thalamus by the matrix cells, there's some, I don't know, something that represents something that makes these cells cascade like this, and that thing can get modified to make them cascade slower or faster. And so all you're doing is modifying that little, you're changing that little, I don't know what, if it's a cell or chemical gradients, I don't know what it is.
Yeah.
It seems analogous to me to grid scale scaling. Like there's some signal, I don't know where or how it comes out, but you can make the module scaling a bit larger or a bit smaller to some degree based on some kind of signal, and then from there you've just got a denser grid or denser denser grid covering space, or a slightly less dense one based on sort of the size of the environment. I think it would be something analogous here.
It's analogous to that, but in that case, we have a better idea that there's this background frequency that is just going all the time. You can't, it's not like a clock, but it's a frequency, and you can speed up the frequency and slow down the frequency, and that'll change the speed of path integration. So it's similar, not identical, because that's yeah, you know, maybe there's a background frequency inside the thalamus that you're speeding up and slowing down. That could be, I don't know.
Yeah, I was gonna say, because there's like pacemaker cells in a lot of parts of the body, like those are in the heart to generate heartbeats, but they also exist in your brainstem for breathing, and I think for circadian rhythm. So maybe there would be something similar, sort of providing just like a background.
Yeah, I think it's believed that several of these originate in the thalamus. Isn't like the theta one originates in the thalamus. So anyway, this is the model. I'm not making this up completely. I think there's a lot of evidence for this, but it's like, you know, the thalamus has these bunch of background frequencies that it broadcasts. It can speed them up and slow them down. That'll speed up and slow down. That'll stretch dimensions. You know, in space, like Scott was just talking about. And but this timing between elements for like melodies is it's not exactly the same. You know, there's I don't the way I yeah, I don't think so. It could be. I don't know. We don't know enough about it. But,
Yeah, I guess to a certain degree, we kind of have a default scale as well, in the sense that like a column can kind of choose to modify the scale coming in or not.
right.
So by not modifying the scale, like that's somehow like a default. So it's almost
Right.
like if no one is telling the matrix cells, oh, if you're early or you're late, there is some sort of kind of consistent.
Right. They
So
would
then,
naturally do
yeah,
the same timing sequence unless told otherwise. And
which
you're
yeah
right.
feels like
It's
that should make it simple to implement in Monty, because then we basically just have like a baseline kind of shot clock, whatever we want to call it. Behaviors are tied to particular points, to time points, and then but we can always contract time or expand time as like a feedback signal.
a shot clock. See what it is.
Yeah, I think
Um,
that makes it a
It's a
lot more
countdown
practical when
timer.
you have to implement.
Right.
but it's not counting down to something.
Yeah, it's counting down to 0. So
Isn't
Does it?
it's
Does
It's a
called
it
countdown
shot
want
clock
timer.
to
'cause in
reach 0?
sports, it's in sports, you have a certain amount of time to make a shot, and if after that you lose it. So
Oh,
so
sorry, but I mean in the brain, it's not like we're counting down to 0.
no, we're counting up, but it's
Yeah.
So
but
in
my
that
point
sense, it's
is
more
it's
like
not
a stopwatch
it's
that
not a clock that
starts.
continues. It's a clock that goes for some duration and then it's done.
Yeah,
And
maybe like a stopwatch that is reset with every event.
right. But it only goes for it's more it'd be like a stopwatch that only goes up to 1 second and then it
Right, a
stops.
stopwatch made of radioactive material that decays after you trigger it.
Well, it's like, you know, in the case
Disintegrates.
of melody, if what Dan Levitin said, it's, you know, you got up to about a second.
What about one of those
That-
sand timers? Has a limited amount of sand in it.
Oh, yeah. That's a long clock. Anyway, so I think it's sort of a shot clock is pretty close. It's not a clock, and most clocks just continue on and keep track of time. A shot clock has got a set duration, it's pretty short, and then it stops. So in that regard, it's like a shot clock. Now, it's a whole separate question: is do we want to emulate this in Monty? You know,
I think some of the things are really nice, like having a global thing that the other parts speed up or slow down. I'm guessing that
that
brings
would be
advantages.
essential, I assume, because you got every learning module has to stay coordinated, right?
Right.
Yeah.
So like something like that, and then in some sense, yeah, like we could make it have like an infinite global clock, but by maybe enforcing it to have like some, or at least try and like constrain the durations, then we force it to kind of have a more sort of local relative notion of time, which is probably gives robustness in terms of recognizing things. And because again, it's a bit like kind of translation invariance we get because all of our representations are in relative spatial coordinates. Monty could have like a single global coordinate and it learns everything in that space, but that wouldn't work well for things like translation invariance. So it feels kind of similar with time.
Well, the thing about it here, it seems that you want to emulate at least some parts of what the brain does. And I think we unlikely to find any kind of global clock that says, you know, starts at midnight and, you know, goes to 24 hours and goes again, you know. So that's unlikely. And there's no, you know, there's no like universal time from, you know, the origin of the universe type of thing. And so it seems even these time cells seem to be sort of duration timers, right? They're just sort of marking the time from one event to some other event, and if you have it in between events, it probably gets reset again. So that seems pretty important. But we could also, if we wanted to, you know, add features that brains don't have and have some other kind of clocks. But, you know, I don't know, you know, universal time or something. I don't know why you would do that, but
Yeah. So I think in terms of the way this curve works and how time cells is sort of the theoretical version. how their time field gets wider as the delay increases is a workaround for computational constraints of the brain more than it is like it basically gives you a way to not entirely run out if that clock gets long. It just means your resolution just gets worse essentially. So I think theoretically we could have like superhuman if we just made this longer, gave us more cells, and gave us
Yeah.
more
Yeah,
vertically.
It
That would be
probably
like
is. It is a
superhuman.
nice form of kind of sparsity almost because in represent because it's probably true that like over longer horizons, precision is less important in order to act intelligently and
Yeah.
change the world. Like whereas over shorter time scales, higher precision is important. So partly it's probably evolved. I mean, I agree. Yeah, it's partly probably I mean, it definitely is kind of just a practical thing, but it might be a practical thing that we also want to adopt.
Right.
Right.
Imagine Monty is having a conversation with a human, right? And the human's got clocks that work like this one. And so the human might have a fair amount of variation in its timing when it gets further out. But if Monty was super precise, Monty might say, "Hey, your speech is wrong. You're off by 10 milliseconds out here or something." It's like, you don't sound right. So
Yeah.
Yeah, like we would
yeah,
want to have more tolerance on the larger time scales as well, not just precision, but also the tolerance.
right. I mean, the nice thing is you could implement Monty. Let's say you had a timing signal, a shot clock like this, that had this stretching out over time. It wouldn't be. You could just change it pretty easily, or change that decay factor and just start over again. I mean, the code wise in Monty would be unchanged, really. I mean, nothing else would need to know this. You just have to train again. I mean, a system that had one sort of decay and another system that other decay would have the same internal mechanisms. It would you'd have to just train again. So it'd be easier to implement and test.
Yeah, and then for now we can, in kind of a similar way to what we're doing with space, keep the system interpretable and kind of relatively simple by representing time explicitly, with seconds or whatever. But then there's the option maybe one day if we feel like there's a strong case to kind of shift to like actual time cells.
Or the other way, you might argue that you do it, with this sort of shot clock mechanism, whether it's cells or not, it doesn't really matter. Then you might have universal time, which would be good for logging and for recalling past events, or searching history and accountability for mine. If you ask me like, "Oh, what day did you go and see the zoo?" And I go, "I don't know, sometime maybe a year ago or 3 years ago."
Right.
I can't remember, last week, I don't know. Whereas, Monty could be like, "Oh, I know exactly when I did that." I have a universal clock that keeps track of everything.
And then I guess the bigger another remaining question is like, I mean, with melodies, it's pretty straightforward what the events are and when it should be reset. But with like more smooth behaviors, like a wheel spinning or a person walking or something like that, it's not really clear what should define the onset of the events and like
Right.
when the interval starts and ends.
Right.
So
I think I wrote that in my notes someplace.
Yeah, I was wondering if like it seems with this neural mechanism, it shouldn't be too big of an issue to associate intermediate observations. Like basically, if we think of these neurons as being like as a cascading activity throughout that interval, whenever some neurons are active in or send projections to layer 1, the apical dendrites could pick up any point in that interval and associate something with them. And then it could just be reset at arbitrary intervals after some time has elapsed. And that way we could represent like continuous space in time.
Well, the problem is the arbitrary, it's like.
Yeah, I guess like
I'm not sure I
max
followed
intervals
the.
changed.
this is similar so the question was, this mechanism that we've been talking about works really well for like things like melody. But the sequences that don't progress, they're cyclical, or they don't have any obvious events. Like walking. If I see someone walking or I'm walking, I can recognize if they walk a little faster, a little slower. I don't have problems with that. But there are no obvious points in time where you'd start the shot clock. It's just like a cycle. And we had the same problem when we talked about melodies. When we were doing melodies with the temporal memory algorithm, there were certain patterns of like events. If they just cycled through, it would be no beginning and end to them. And so the temporal memory algorithm would fail. If I have a pattern like A, B, C, D, A, B, C, D, A, B, C, D, A, B, C, D, and I just do it over and over and over again, the temporal memory algorithm would try to represent every new A and as unique from every previous A. And it was as if it was one very long high level or high order sequence. And so we never really figured out how that worked. And this is similar, right? We have repeating sequences of no events.
I guess is that partly resolved through hierarchy? So like if you have a sequence, as long as that's represented kind of through pooling as like a single representation, then I mean it's still repeating, but then at least
right, but then I'm repeating the sequence over and over again, and that becomes its own problem.
Yeah. But at least it's maybe simplifies it, the representational space.
Imagine someone's moving, you're moving or you're seeing a dot moving around on a circle. Right? Okay. Now I could speed it up. And it's, oh yeah, that's, maybe your job is to recognize dots moving counterclockwise versus dots moving clockwise, whatever. I don't know what it is, but the point is there's a very simple sequence. You can learn to predict it, and yet it's repeating over and over again. It's not like a melody. It doesn't begin and end, and there's no obvious point in which you would say, okay, what's the tempo? Where do I mark it from? So how do I represent the speed of that dot moving around the circle? I can't use it using the shot clock mechanism. I don't see how.
Yeah, unless the shot clock is kind of constantly restarting for each of those subsequences.
Yeah, but when does it restart? At the top? I don't know.
So yeah, I think maybe a partial solution could be, or maybe, so I have two parts of the solution, maybe proposals. One is we could, well, maybe I start with the other one. Could you go to the slide 3 again, Scott?
This one?
Yeah. So let's say let's assume as the circle spins around once, we kind of go through this, can I point somehow? I don't know. We go through that, like the single trial responses plot, the red line, we go through that.
Can we zoom in?
Yeah. And so let's say it spins around once and it never resets because there was no discrete event. We're just going down that diagonal. But still, whenever any of these neurons are active, we can make associations in the column with whatever sensory input is coming in at that point in time. Like, it's not like we can't make we can only make associations at the 0 point or at the beginning of an interval. We can make associations along this entire diagonal with those different neurons. So we can.
But we have to have the sequence the shot clock has to start at the same point, right?
Yeah, we're kind of like
so
the period,
the
period of the whatever behavior is, like walking, should be the same as the period of going through these. Otherwise, it's going to keep shifting, and we're going to associate new cells every time.
Or you could have
just
multiple
like obliterate.
shot clocks during. You could have multiple shot clocks during a period. Imagine I was doing a dance, and often on a dance you might have a heavier foot stomp on the beat, right? And so you go bump 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 bump, and so that louder beat is would be something to start. I think even Vivian, you mentioned a while ago some metronomes, not the old type but modern ones, they'll like have a beat, let's say, tick, 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 but then on every fourth one, they make it a little bit louder.
Yeah.
You can imagine that's like, okay, that's the one that we want to start the clock again,
Yeah, so that's kind of what I'm getting. I'm trying to break the problem into two problems. One is when do we reset this clock? And I didn't really mention that yet. The second one is storing kind of continuous events between the resets, because there's a lot happening between kind of the behavior starting over, and there aren't really discrete
right,
events.
I see. You're just saying in terms of storing, you'd have the information you want.
Yeah, exactly. So like, even though there are no discrete events as the circle is spinning, we could still store all of that.
So maybe, I mean, it's
then the second part of like when we reset it is I was thinking of as a separate problem, since yeah, for sure we need to reset it and we need to have some kind of signal for that.
right.
And what we could use for it maybe is like recognizing specific states, even maybe morphological states. Like maybe you've seen the circle before it started spinning, and you learned with the dot at the top. And whenever it's in that state, the morphological model of the wheel is being recognized again, and that kind of triggers like setting us
Maybe.
into the beginning state of the sequence again or something.
Yeah, or like walking. I feel like if you were going to draw someone or think of someone walking like this, maybe the spread legs kind of like this is almost like the default, because it's kind of it's simpler than like all these other complex positions that people, and so it might just be like you need something to like anchor onto, and
Right.
yeah.
And I mean, there might be multiple ones that just kind of anchor
Right.
us to different states in the sequence. And
Right.
Yeah, I don't know if anyone else, this is a related sort of fun observation. I don't know if anyone else has noticed this, but I just realized there's something I do all the time, which is related to this. If I hear like the clock in my kitchen, it makes an audible click every second. Most of the times you don't notice, but if you're sitting there and it's silent in the room, you hear it and I can focus on it. When I do that, I almost always hear every other beat sounding louder than the previous beat. It doesn't feel like they're equal, but I know they're equal. So one feels like a little bit louder, and then so my brain. Well, my point of this is my brain naturally wants to assign a beginning to something. It just naturally wants to say this is, let's mark a spot here, even though there actually isn't true. So I wonder if like when you see someone walking, your brain will just pick something like you were saying, Niels, like, oh, maybe when the left foot goes down, that's what, that I imagine that person going 1, 2, 3, 4, 2, something like that, even though they're really just going ding, 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 ding. So there might be some fabricated events. The brain
and then I
just
guess
might wanna
another thing
pick
that could
one.
help, another thing that could help is like some of these sequences, like I'm just looking at a fan that's spinning right now. Like some of these sequences, I'm not sure we'd even really learn as a sequence. Like we've talked about how like people didn't really know what horse galloping looked like until the camera was invented, because there was no way to kind of have that morphological model. But so some things like this, you could almost store more as just like the feature input is a flow pattern. And the model is basically just like, oh, I expect that flow pattern, but I don't model it in like oh, I know this happens and then this happens and then this happens.
Well, I think interesting. The fan is a good example of that. Although I would argue the horse, what they didn't know was whether the horse's hoofs are all coming off the ground at the same time. But I bet you there's a noticeable cadence to the horse when it's galloping. That
for sure. I mean, you could definitely recognize it, and you can definitely recognize when it's wrong.
No, but I'm saying you'd also recognize like there's probably a point where the horse's body goes to highest or something like that, where you can see there's probably some demarcation, unlike your fan. There's probably some demarcation where you could say this is a point in the gallop that repeats. So we have things like melodies where there's no repeat of the elements. We have things that do repeat, but cycle over and over again, like the dot on the circle. And then you got something like your fan, which is really, if I look at my fan, I generally can't see the individual blades when they're going around. And so it's something else. It's just like, it's a movement field, vector field of some sort,
Right.
So, and then that, I'm not sure if the timing, I guess if I took away the sound from your fan, could you tell if it's going a little faster or a little slower? Interesting, you know.
Yeah,
If you
it'd be
can't,
hard.
then maybe the time's not involved at all.
But if it is going slower, you do start to kind of see the blades, and
Well,
then
at some point, yeah,
that's
then
like
your visual perception changes. So anyway, we were defining a set of tasks. I just tried to enumerate the 3 we've talked about so far.
And so, yeah, it seems like with almost all of them, we can pick significant points in the sequence to use as kind of reset points of the time intervals.
Okay. And just state it again, if I see the dot going around the circle, I think I might have a perceptual distinction when the dot gets to, say, the top of the circle. I might mentally just divide it up and like, oh, we're back to the beginning type of thing, you know.
Yeah, often there's like often there are some kind of ways that things align. Like, for example, if you imagine a little arrow at the top and the dot is another arrow, and then whenever they align, your brain kind of snaps it.
Right.
Like,
Well,
this is a lap and this is another lap.
Right. Clearly, if I had a little stationary dot and a moving dot, wherever the stationary dot was, anywhere on the circle, I would mark that as the beginning and the the loop and the end of the loop.
Yeah.
If I didn't have the stationary dot, all I had was the moving dot, I think I would naturally pick like the top or the bottom. I wouldn't pick something at 30 degrees or 27 degrees, you know. That just doesn't seem natural to me. I would be like, oh, there we go, back to the beginning, ding, ding, ding. Like if I were to have to say a sound every time the dot goes around, makes a complete circle, I would almost certainly pick that. Just naturally, I'd pick the sound like at the top or the bottom or something. I would pick a spot and then watch it get back to that spot, which is sort of indication that we actually do maybe make up an event.
Yeah, I feel like this happened a little with Rami's thing, because wasn't that just kind of cycling infinitely? But then, sorry, this is the exploding logo.
Oh, okay. I was gonna say, which is Rami's thing?
Sorry, the exploding logo. But then I think it was cycling infinitely, but then, we had seen it start, and that just kind of naturally felt like the starting point going forward. And so it felt like whenever we got to that point,
it actually the beginning is when it explodes, right? It's a small logo and then it goes boo.
Yeah, I guess I guess it is a simpler one in that sense.
It's simpler. Yeah, I guess if all the 4 parts were just randomly, had random start positions and never came back into the logo, it might be a little more difficult. But it's a good example, because I think if I just took some randomly moving things like that, as long as they're all moving together, right? They all have to have the same period. If they have different periods, you're hosed. But if they have the same period, there will be some point in that sequence where you would your brain would say, okay, they're the smallest, or they're the biggest, or they're all horizontal, or something.
Right.
That you can easily recognize.
And I guess, yeah, the less that is available, the harder it is to learn the sequence, which is why it's so important in music to have the kind of consistent beat and stuff. Because musicians, people dancing, everyone's going to get confused if you don't provide that kind of signal.
Right.
Right. And so our brain can kind of try and get by without it, but clearly it's better at modeling sequences and behaviors if it's available.
It got a little complicated. Viviane brought this up. If you play music, everyone who knows this, you always are hearing the beat, and it's natural to tap your foot, to have a physical, marking of time, and that of course is not a duration. That's like it's like a start clock over and over again. But and of course, if you play in an orchestra or something, you have to practice not stopping tapping your foot. You're not allowed to tap your foot. So you have to just mentally hear it. But that complicates things a bit, because I don't know how the interaction between the duration of a note and the background beat, because sometimes notes go before the beat, sometimes after it. Is the shot clock starting on every tap of your foot, or is it? I don't know. That's a little weird. I don't see
Yeah,
that.
maybe it helps reset the shot clock, and then you have the relative place of the note relative to the beat.
Well, so that would, yeah, so if it's relative to the beat, right? Although sometimes it's right before the beat, right? You have a pickup
Or
note.
as in like, yeah, because could you not just have like multiple kind of shot clocks, and so some of them it's basically different sets of time cells. So some are more precise, shorter duration, and some are longer.
Maybe.
Maybe, I mean, maybe the beat just kind of helps keep everything consistent in the long term, because I mean, you can whistle a melody without tapping your foot or anything like that. You can reproduce the melody without the beat. You know
Right.
the kind of relative placement of notes, but the beat kind of helps you keep that rhythm in the long term and not kind of slowly get slower or faster.
Right. The beat also might be just a means of setting the slope of the decay curve, maybe, you know. Anyway, I think it's a mystery. I think there's still some unanswered questions, but I don't know if we really want to try to solve those today.
Yeah, one fun fact I came across, I think I mentioned it in the comment on the document, is that apparently most non-human species don't have beat perception. Like they have tempo perception and like temporal grouping, but not beat perception. So it's maybe not really a crucial mechanism to understand for
How are
human
those different?
behavior. Um, Oh, I didn't read the paper that much in depth because it didn't seem that relevant, but I think they can do like they can learn time intervals very precisely, but like if you play a melody, no, what was it? Um, they can't reproduce like they wouldn't be able to tap their foot or nod their head or something to a beat or
Right.
reproduce something in a certain
I mean,
tempo over and over again.
all mammals have this, and this is a common test, right? Where they do some sort of delay thing where they have the animal sees a stimulus and they have to delay a certain amount before they react. That's like a one-off type of thing. So it's interesting if the ability to like represent the rhythm and the frequency of notes in a melody is something that most animals don't have, that would limit their physical abilities. You probably wouldn't be able to generate speech. You probably wouldn't be able to generate a lot of detailed physical movements, you know, that we can do. It would be pretty limiting to an animal. I mean, it's interesting that it could come along with humans having language, you know what I'm saying? It's a requirement for language.
Yeah, I think there were a few species they mentioned. I think one of them was a parrot that can do it. Like, you know, those videos of the parrot nodding its head
Yeah,
to
right.
music. So and then that one does some kind of speech in some cases without too
Right.
much meaning, but yeah.
That's kind of interesting. Yeah.
So there's been some debate about this, and I went to a talk by the guy who works with the parrot, and the debate was sort of about whether um, we only see it in certain animals because they have some kind of intrinsic reward for it. Like we get some intrinsic reward for, you know, tapping a beat or dancing or whatever. And so the question is whether or not maybe every mammal does have this ability, but we just don't see them exhibit it because they don't get any joy from
Well, you know,
dancing or whatever.
that's
You know?
weird you say that, Scott, because when I was a kid, music was like this huge mystery to me. It's like, why the hell do we enjoy this? What is the benefit? You're saying there's intrinsic. I couldn't figure out the intrinsic benefit of music. It was like you just wiggle these air molecules around, and all of a sudden your whole mood changes and you start doing weird stuff. Like
Yeah.
it made no sense to me. It's like, what's the reward for like, wow, that's a great song? You know, I don't know. I mean, take the lyrics out, you know. So maybe there's an intrinsic reward, but that wasn't obvious to me. It was like, what the
Yeah.
hell is it?
In my old PhD lab, we actually tried this. We used genetically modified mice so that we could excite their dopamine with a fiber optic cable. And
Yeah.
then we tried to teach it to rear up and down to the beat by like if it reared up with the beat, we'd like give it some dopamine to try to like and give
Wow.
it that reward, you know, to see if we could get mice to dance. Basically, the project was called Tiny Dancer. Yeah,
Did it work?
and I think it was inconclusive. It was
Yeah.
inconclusive. Yeah. Like there was some evidence for it, but I
That's
think a
amazing.
lot more needed to be done to make it, you know.
Do you still
Our
have the grant,
research,
But, yeah.
the grant
our
request for that?
research
I'd like
dollars
to.
at work.
Yeah.
Great. Oh, that's fun. All right, where are we?
I mean, it sounds like I hadn't read all the comments on your document, Vivian, but it sounds like we've made some good progress on the open questions.
Yeah, I think
I-
I have a much better idea now how I would implement it in Monty. Now I'll plan to update the document with that. I guess I'm still not 100% clear like how Monty would kind of make up events to restart the clock, or like how we would define them. But I think there will be ways like using morphological models when they're recognized, or something like that. Or
Yeah, it feels a little bit almost like how, you know, we talked about how like how would you recognize that some mugs have handles and some don't, and kind of form separate models. It feels like maybe if a column's just constantly trying to build models, like there'll be it'll be challenging, it'll get brief glimpses and stuff, but eventually it might kind of coalesce on like, okay, this is, now I'm voting on this. This is a state I recognize. Like, you know, the person's like, yeah, either legs are spread or both their feet are on the ground or whatever. And then once that's kind of a thing that can appear when that representationally, when it appears in the world, then that gives you something to kind of anchor on, and then you can start actually kind of predicting the sequence. So it's almost like you would need to build a model first that you're going to anchor time to.
like do you mean like you might learn a longer model at first and then you would notice that it's actually repeating itself and forget part of it again?
Yeah, maybe kind of like a blurry model of everything. I guess I was just thinking like, yeah, at first you wouldn't really be able to predict any state, but like eventually you might be able to then be like, okay, now I recognize this brief point, and then but then recognizing that it repeats kind of comes later. It's more just like that you recognize it at all as like a state, but you learn to recognize it because it does repeat.
I have it's just along the same lines, Niels. I can just speculate on ideas about this. I think I didn't follow everything you said. But I think we're talking about the idea of like, well, okay, if it's not obvious what the events are, what do we do? A couple thoughts on that. First of all, why do events exist? In music, for example, if I'm learning a melody, essentially new notes are unexpected. That is, I don't know the melody yet. So when a note occurs, like, oh, here's something I didn't anticipate. I mean, I couldn't predict a note until it happened. And that would lead to a bursting of cells, and then bursting of cells in the column. And then brief burst, that's an event. And you could use that as your trigger. And you can say, okay, something just happened. Let's remember this time. So that's the other case. That's the case where the events are very clear. Well, what if the events aren't clear? You've got something moving in a circle or something or whatever. It seems like the matrix cells, this is all purely speculative. The matrix cells could be wanting to be triggered. They don't want to sit there idle, right? They want to do something. They're not useful if they're not firing. So they might on their own have make it more easily triggered. That is, they might say, the closer you get to your end of your time duration, they might take less of an event. So you might try to pick up a subtle change. I don't know what that would be, but the point is they could be looking for less obvious events, or they could even just start oscillating on their own. They might just, like a background rhythm. They might you might imagine just you're hearing a beat in the background, and they're I don't know. I think that I guess what I'm saying here is there's ways that.
Yeah.
Go ahead.
That's what I was trying to get at earlier, that they might just, like, as they get towards the end of their representational capacity, just kind of restart, even if there wasn't an event.
Right. Well, they'll
But
may be easy to restart. They may be like, okay, I'm gonna pick up this one. Anything.
Well, and I think, I mean, yeah, what I was kind of saying earlier, I don't know if that made sense, Vivian, that I feel like it's important that at least eventually the period of the cells needs to match the period of the behavior. Otherwise, like we're going to like what cells we're trying to anchor behavioral states to are going to constantly be shifting. But I guess what they start with could be arbitrary, kind of like you're saying. And then we're trying to predict things. And if we're failing to predict things, then we would try and predict earlier or later. And so that's going to slowly modify the period that they fire to. And then I guess eventually it might match the period of the behavior. And then we can actually like lay down those Hebbian connections strongly that tell us like, okay, yeah, now predict this, now predict this.
Well,
So
melodies have a beat, right? But you know, behavioral sequences don't. I'm sorry, I'm just thinking out loud.
So in what you're talking about, Niels, are you talking about an example where the whole behavior fits into one cycle, into one interval, or there are like multiple different length intervals?
Yeah, so
Like
it all fits into one
something-
interval, like walking, but we don't necessarily know where it starts and ends.
Yeah.
And so like, let's say cell 2 that tends to fire early in the sequence, like if the sequence, if the timing between when cell 2 becomes active again is not aligned, isn't the same period as the behavior of walking, the behavioral state of walking that it's kind of assigned to, that it associates with, is going to constantly shift. And it will, like, we won't be able to learn a meaningful association. But maybe we can kind of tweak that period over time as we see the behavior based on like how well we're predicting the sequence or not. And so basically, like you're saying, at least at the start, it could be arbitrary, and then it can kind of refine itself.
Yeah.
But yeah, it's a bit of a
Another thing that-
bootstrapping thing because it's like, how do you refine your prediction until you're making predictions, and how do you make predictions until you've assigned stuff? So it's
Well,
it feels a little like kind of yeah, it feels a little like SLAM or something. It's almost like we're trying to like build a map of time, but we're also trying to associate where we are in time. And it's kind of maybe like an iterative thing where we can improve the map of time, the period of the sequence, as well as where we are over.
But is there
iteration.
are we trying to learn
But when I say map of time, I'm using that very loosely. I'm just trying to make an analogy to SLAM.
I think it's
In terms of it being a bootstrapping process.
If we assume that the shot clock resets on events, and then assume we always have some sort of event, then we don't have a problem because the shot clock has its default decay rate, and we'll learn whatever we learn initially will be on that default decay rate, and so we're all good. The only problem is, I think, maybe I didn't follow everything you said, Niels, but I think the problem we're trying to deal with, what is if there's not an attack? There's no obvious event in which to start.
I think maybe we're talking about two slightly different problems.
Maybe.
So as far as I understand, like, yeah, there's the problem of like there are no events versus there are events, but there, even if there are events, there's still the problem of when does the sequence restart? Like if I have ABCD, like
That's not a timing issue, that's a sequence memory issue.
Yeah. And I think maybe Niels, the solution you were talking about, as far as I understood it, got a bit more at like how do we know when the sequence ends and we need to start at the beginning again, which is similar to like when do we like chop continuous behavior into parts.
Yeah,
But maybe
yeah. I mean, I think it's overlapped with if you have a continuous behavior with no obvious state, like trigger.
Well, there's two things. One, you have a continuous behavior, even though there are events in it, A, B, C, D, A, B, C, D, A, B, C, D, that you don't want to learn as a high order sequence. That's one problem, but that's not the
Yeah,
same
that
problem
wasn't
as,
actually what I was trying to address, but I guess it is related. I feel like maybe I think having the appropriate periods could maybe solve both. Because but it's less obvious to me how you solve the problem you're describing, Jeff, because there's more to it than just that. It's like, how do you decide that you're going to model it as a repeating rather than a higher order sequence? I don't know. Maybe
Yeah,
just kind of sparsity, like, you can
well,
try
anyway,
modeling it as a higher order sequence, but you fail. So you
well,
eventually you kind of realize,
yeah.
okay, I need a more constrained representation.
we never resolved this issue. It was hard.
So no
Good
pressure
news. Yeah, I
on
think I understand now more what you were getting at. I think the reason I thought of like when does it restart is because I think the issue you were pointing out doesn't happen until the behavior restarts and then things are unaligned anymore.
Exactly.
So, okay, cool.
I'm not following that, but that's okay.
Yeah, maybe I can try and draw something for next week, or just a post on Slack. It might help.
Aren't you guys on your offsite next week? When's that?
The
2
week
weeks
after next week.
Oh, it's 2 weeks.
But yeah, I think generally we may, at least in my mind, it's much more concrete. And yeah, we can think a bit more about like how do we know when the sequence restarts and how do we define events when there are no clear
Yeah,
events.
those are right. We don't have a way of even representing a repetitive sequence that just goes on forever. We don't know the neural mechanism for that. And we have to solve the problem of inventing events. I think inventing events it doesn't bother me. I think it seems like we can come up with a bunch of ways of doing that. The infinite loop is a trickier thing for me.
Yeah. Okay, I wrote both of those down and we can think about them some more. If you like, I can still talk a bit about the matrix cell literature because I feel like
Sure.
maybe that ties in nicely on like one, the connectivity between matrix cells to the cortical column and where the state could be represented, and then also like whether this is actually the place that's most likely to implement the global clock, which
Yeah, sounds good.
long story short, I didn't find a more likely place. So but yeah, just as a brief intro, because I didn't know much about matrix cells at all, so I just wanted to read about them and maybe it's interesting for everyone. So basically, this was, I think, like one of the big papers proposing this first was from Jones in 1998, where he basically divides the neurons in the thalamus into two broad categories. Core cells, which are basically like the relay cells we always talk about. They are topographically structured. There's a precise stimulus response pairing, and they have very precise receptive fields that can be easily characterized. And there's a precise projection from nuclei to specific target regions. And the projections go to the middle layers of the cortical column, so like lower layer 3 and layer 4. And the way he mostly characterized these two is by looking at their immunoreactivity to different calcium binding proteins. So this one is the core cells were reactive to parvalbumin, whereas the matrix cells were reactive to the 28K calbindin. So they seem to be having like also some different chemical properties. And then the matrix cells were found spread across the entire thalamus, irrespective of the nuclei. there were a bit more matrix cells in nuclei that had no core cells or like no direct sensory input. And
By the way, I'm not sure what nuclei would have no core cells. That's weird.
yeah, I think it's a bit of a relative thing. So yeah, here's a plot of the core cell distribution in the different nuclei of the macaque thalamus, and there are some here. I'm not sure what I can pull out the paper what they refer to.
It's okay.
H-
It just it's a in our theory, all the nuclei should have relay cells. So I don't know what's going on if they don't, or if they just didn't identify them. I
yeah,
think Jones, if I recall, he really used these Pavlov-Buman and the Kalbinden methods for identifying these cells, which to my mind is a bit suspect because that's just an association. You don't really know if that's really always the case.
yeah.
So there might be thalamic relay cells that don't have Pavlov-Buman receptors.
Yeah, exactly. So yeah, he relied basically just on the different
Right.
properties of these cells to characterize them. And actually, I found this pretty recent paper from Sherman and Usraid that says that this kind of classification is too simplistic and basically
All right,
say, yes,
so-
there are clearly core cells, but there's no clear evidence for a single class that encompasses all of the matrix cells, and there's great variation in connections made by the
Right.
thalamic projections,
Interesting.
fit neither core nor matrix classification. And basically that it often leads to confusion because there isn't really a clear mapping between like first order and higher order nuclei and matrix cells. And yeah, that
interesting.
basically
So
this is a bit too simplistic.
I know we're recording, so I'll just keep my comments pretty high level here, but back in the day, there was two major camps of researchers. There was the Jones camp and the Sherman camp, and they weren't really pals. And so they use completely different language for describing the same thing, and you're seeing that right here, right? This is Sherman saying we don't like Jones' language, and Jones ignores what Sherman uses, and so as you read these papers, it's very difficult to piece this together. I'm surprised you found this here. This is a great little quote. Anyway, so it does make things much more complicated when you're reading this literature. When I read the literature about the thalamus, I immediately say, oh, this is a Jones paper. Okay. This is a Sherman paper. Oh, that's really different. Personally, I found the Sherman descriptions much more to my liking because Jones would make this distinction about these receptors, just like Sherman's pointing out here. But it's not clear that that's an important distinction at all. It's just like some cells look like this, some cells, okay, but what do they do? What's the purpose of that? There was never a connection between these binding cells, these binding proteins, and what the cells actually did. So I always was take Jones's papers and have to say, okay, be careful here.
Yeah. Yeah, so
We ran
definitely
across
it's
that.
not as clear cut of a view as I showed here. But yeah, basically the idea is that these matrix cells don't really care about the nuclei boundaries, and they project much more widely to the cortex across different topographic areas, and their input is more imprecise and less topographically ordered. They have harder to define receptive fields. And yeah, they project widely to the superficial layers. I think, yeah, here it's shown too. And then also they project to mostly the upper layers in cortex, so layer 1 and 2 and maybe upper layer 3. So yeah, this is kind of this is from the original Jones paper, and then they did another review paper like 3 years later with the figures looking more colored. So I'll show this one now. But yeah, basically here's some examples showing that, yeah, the matrix cells this is BPN, which the matrix cells project to the somatosensory cortex, layer 3, 4, and then the core cells project to somatosensory cortex. And then the matrix cells are basically everywhere and get input from different sources and project to the upper layers. Here's auditory cortex.
Are they saying that the spinal tract is projecting, is innervating the matrix cells? Is that what they're saying?
Yeah, I think that's what they said
Okay.
there. Let me go to the original figure.
Maybe some pacemaker cells. I don't know.
What was that? Say it
I
again?
said maybe some pacemaker cells,
Mm,
but I
I
don't
don't
know.
know.
What's interesting?
Well, right, the key thing was for this matrix cell theory to be correct, we had to see a projection from the cortex back to the matrix cells.
Yeah, so I got to that in a second.
So this subcortical input to the matrix cells, I don't know what to make of that.
Yeah, I have a nicer diagram about that in a moment, but just to briefly go to the other modalities. So here, this is the auditory nuclei, where we have the direct auditory pathway that goes directly to like the primary auditory cortex, but then the matrix cells project much more widely to neighboring regions as well. And then here's LGN, and basically we have the magno and parvocellular layers that project to layer 4, and then the matrix cells are basically everywhere, including the S layers, they call it in interlaminar zones, and then project to these upper layers. And so I promised a better picture. So maybe this is not the best picture. Well, basically let me see if I have maybe this one is better,
Rolling consciousness, wow.
even though it says conscious content. But yeah, basically this shows that the matrix cells project to the apical dendrites of layer 2-3 cells, but also layer 5B cells. And then layer 5B cells project back down to the thalamus, both to core and to matrix cells. And I think that was
Say
the
it again.
main,
Say who projects down the matrix cells?
the layer 5B cells.
Layer 5B cells.
Yeah, let me see, I've added
This
some
is in
quotes.
monkey.
Yeah, both matrix and core thalamic cells receive driving inputs from layer 5B cells. I think TT is thick tufted, maybe.
I'm not sure. Is this a layer 5B? Is this the motor output cells or this the other ones now?
Those are the other ones, I think.
Yeah, because he has a
So-
picture of a monkey on the right there. So
Yeah, I think those are not the motor. Let me go back to that paper. Well, this was a review paper, so it could be anything. See if they say it in the caption. So A was
there's dendrites of TTL5B cells in L layer 5A. What the hell? Okay.
yeah, so this is this little purple arrow here that
I
the matrix
see.
cells don't just project to layer 1 apparently, but also
Okay.
some
Yeah.
dendrites from those layer 5B cells. But what I was kind of thinking was that when we have like overviews, we also have the voting connections between, like lateral connections between layer 5B cells. So that potentially we could represent like the state and time in layer 5B, and that's also something that can be voted on. And it would kind of match nicely with location in space being in layer 6A and then location in time being in layer 5B. And I'm not sure if it matters.
I don't know why you'd have to vote if everyone's getting the same signal. There's no reason to vote.
Maybe the state in the specific sequence instead of just the duration of the interval.
I don't think it could be, but I never thought that the matrix cells would represent specific sequences.
Oh, not the matrix cells. I mean, the layer 5B cells would
Oh,
represent the
Right.
state in a specific sequence, whereas like the input from the matrix cells is just the
I
duration
see.
from the
Oh, I'm
current
sorry, all right.
interval.
Yeah, that's interesting. I'm also trying to think of like if it was the same projection as the motor output, how it would work in terms of the matrix cells receiving that, and then knowing that the signal was too early versus too late.
I don't think it's the same projection as the motor output. I think that's the ordering where layer 5A is the motor output
Yeah,
projection.
okay.
Right.
But it is weird because or at least, yeah, I wasn't aware of like another major output from layer
Well,
5
I mean, the layer
like
5B
that.
cells, no one has any idea what they do, right? So we've been speculating lots of different things. We don't know. But don't go on here because there's that note D on that figure. It says sparse bursts, green color occur in both conditions, but are time locked to stimulus onset of hit.
Yeah, so
Stimulus
that's
onset is your event.
yeah, exactly. I thought that was a nice.
That's a big clue. I mean, that's what we want to expect, right? We want to see that the matrix cells become, I don't know what the hit and a miss in this case is, but when it says stimulus onset, that's the event that you want to start the clock. So that's consistent with this theory.
Yeah, exactly. So
And the
yeah,
hit
it
thing,
was
I wonder if that's basically saying that if they, presumably that miss is they fail to predict things. So basically, if their clock doesn't start at the right time, then they
Well, if the
mess
event
up.
doesn't start at the right time, right?
Yeah, or their representation of it. I'm guessing it's a trial where they have like two different, or like where something happens and they have to respond at the right time. Something like
yeah,
that.
so it's a whisker deflection threshold detection task. So synchronous bursting in layer 5B was shown to be time locked to stimulus onset and also to distinguish hits and false alarms. So trials on which the animal reported perceiving a stimulus. From misses and correct rejections, trials on which the animal didn't report perceiving the stimulus as present. So maybe to frame it a bit, this paper is about how that could contribute to consciousness, because there's actually it's kind of interesting, like both evidence that you can reverse anesthesia by stimulating the thalamus, and that you can trigger kind of vacancy, or also if there's a lesion on both sides of the thalamus, you go into a coma. But basically that when the animal is anesthetized, there's mostly local activity, but then if the thalamus is stimulated, there's this more global spread of information and synchronization of activity. And what was I getting at? Yeah, why the hit and miss trials? That it kind of modulates the threshold at which you become conscious of sensory perceptions.
Well, I mean, when you disable the thalamus, are they disabling all of it? Like both matrix cells and the relay cells?
Well, this was, I think, other research data said coma is associated with lesions that affect the brainstem. However, bilateral thalamic lesions and lesions that decouple the thalamus from the reticular activation system can also cause coma. And
Well, I
recovery
mean,
from coma is associated with improved function in thalamocortical circuits.
right. But my point is, if you were to somehow disable all the relay cells, then basically the brain would be disconnected from anything, and nothing is going to work.
Yeah, as far as I could tell, they didn't specifically identify that it's just the matrix cells.
Right. So it's it's always been obvious to me, like if everything entering the cortex goes to the thalamus and you take out the thalamus, well, then you're just then the parts of the cortex can't talk to each other. at least, then of course you're going to be dead, it's like nothing's going on.
Yeah.
You can't think even. I mean, not only can't get input, but to the extent that connections between cortical regions through the thalamus are essential, then those connections will not be working either.
I agree. Yeah, I was trying to read this paper not through the lens of consciousness, but
Yeah.
like the theory that provides a time signal, and I think a lot of the evidence they summarize there can be interpreted in that lens as
Right,
well.
right.
So I think it's a pretty interesting paper. I can share it later.
Yeah,
But
it looks really interesting. I guess, yeah, what I was trying to, and maybe I misunderstood, but I guess what I was just trying to get at with that hit and miss trial, it's like we were talking earlier about like how do you know when to start the shot clock or whatever? And it seems like what they're observing here is, yeah, that's kind of like an inference problem. And sometimes you get that wrong. And when you get that wrong, your behavior is impaired and you don't do the right thing. So it's like they observe on the trials where the animal's behavior was correct, they seem to observe the shot clock starting when you would want it to start. So it's kind of like, oh, those must be trials where kind of the animal inferred the start correctly. But
I
at least
mean,
based on
I
how I'm
don't,
understanding it, there's other trials where they got it wrong, and
right,
then
well, the
it doesn't
details,
sync.
there's details of this experiment are critical here, because why did the animal, it said it didn't perceive the input maybe. Well, why didn't the animal perceive the input? Was it would they make it dimmer and dimmer or harder and harder?
yeah,
Because if you don't perceive it, meaning basically that means you didn't burst and you didn't start the clock. You basically didn't notice an event occurred.
yeah. So that's what they made it hard to perceive, I think. So following the onset of the target stimulus, a surround pattern appeared, rendering the target stimulus invisible with a probability proportional to the density of the pattern. Monkeys were trained to report the disappearance of the stimulus with a lever press.
So it harder and harder it gets, then at some point you just don't you aren't able to physiologically detect the change, and then you're not going to get the event. So basically in that D figure there, if the upper one's basically saying the animal wasn't able to perceive anything, well, no clock's going to start. As soon as they could perceive it, then bingo, you got your clock.
Yeah. Yeah, and then another thing they show is that the kind of sensitivity to the stimulus intensity is higher or is lower when the thalamus is lesioned, or regions in the thalamus are lesioned. So the way I was kind of thinking of it was that if the matrix cells project to the upper layers and provide kind of the context signal and make it more likely for these cells to fire, and in fact these cells kind of get into a bursting mode if they also receive apical input, that this would basically make it easier for those cells to fire and detect.
But also again, if we're lesioning the entire thalamic region, then your relay cells are going too. And so, so I mean, essentially what the mechanism we proposed back in the sequence memory days was an event would come in. If it wasn't anticipated, you would get a burst of activity. But obviously, if you're not getting input, and that burst of activity essentially starts the clock and says we have to learn, pick a SDR to learn on. Anyway, if you're not getting input, then none of that's going to happen. Not just the matrix cells; the whole thing is going to fall apart.
Right. Yeah, that's true. Oh yeah, one other thing
it's a little bit like
with the burst is
consciousness studies are a little bit like, imagine I gave you a computer and I said, look, there's this thing here. We know it's called the bus. If I break this bus, the thing stops computing. Well, that must be the center of computing. This bus, you get it all wrong. No, it's not. You just broke the whole damn thing. That's all. That's what they do with these consciousness studies. Oh, we broke the thalamus and you're no longer conscious. Well, that's the center of consciousness. I don't know if that's what these people said, but
yeah, I mean, parts of it is also about like differences between sleep, wake, and anesthesia.
oh, that's interesting.
where, like, for example, this figure here, well, this is a simulation, but I think they also have experimental data on this, but basically, so one of the key features of these layer 5B cells is that they shift from a regular spiking, so like this, when
All right,
you
but
have
can I
input
interrupt you
basal
here?
dendrites to, yeah,
I think I remember you right. First of all, if a cell shifts from regular spiking to burst firing, that's the motor output cells. That's the character, and also the TT, I'm guessing that represents thick tufted,
yeah, I think so.
and thick tufted is also the motor output cells. So for whatever reason, we're talking about the motor output cells here, not the other one.
Okay,
I don't know why it's B. I mean, it's who knows what the hell. But those features are the
Yeah.
more indicative features of the motor output cells.
okay, yeah, it's there. Somehow they talk about humans. I'm not sure if it's in that order in humans, but yeah, it's a review paper, so they're not that clear about which ordering. But yeah, if that's what characterizes the motor output cells, then that would make sense with what Niels also said, that those are the ones in layer 5 that project down to the thalamus.
This, by the way, this is really interesting. This is saying one way to interpret this is there's a different firing, this bursting versus regular, which it's been well documented for decades, but they're associating here with simultaneous activation of apical and basal dendrites. In a very simplistic theory that we've been talking about here, that would say when the cell is predicted and it's at its correct timing, it's regular spiking. But if the cell is predicted and you don't have the right timing, meaning you don't getting the layer 1 input from the apical dendrites, then it bursts.
No,
Right?
it's the other way around. If it
Oh, it's the
gets
other way
the
around?
if it gets the apical input, then it bursts.
Oh, it's the other way
If it
around.
doesn't get it, it's regular spiking.
Uh,
So the burst firing is the orange one, simultaneous activation of apical and
all right.
basal
Well,
dendrites.
I'm just wondering if this burst firing, if these cells, if this activity somehow made it back to the matrix cells, that the distinction between burst and non-burst could be the this is how it tells the matrix cells they have to reset the clock. But maybe I got it wrong. I don't know.
Yeah,
That little
reset
detail,
or fire or modulate how quickly they're firing the
right?
matrix cells. Because like somehow we need to communicate that back. It's like, oh, it needs to be earlier or later.
Right. I guess for me, I've read a lot about these cells in their burst and their tonic modes. It's been around for decades. This is the first time I ever read that one's associated with apical input and one is not. That's the first time I've heard that, which is very interesting. It's like, oh, that's a piece of data.
I thought, isn't that in HTM? It's when you burst, then they are. Oh no, maybe not. Well, I guess in HTM, the apical input basically helps you get lateral inhibition, so that you have individual cells, right?
No, we didn't. In the sequence memory, we didn't account for apical inputs. I don't think we did.
Okay. Well, maybe it wasn't
didn't.
apical then, but dendritic input, like a dendritic spike, is what kind of primes the cell.
Right,
so that
right.
Spikes earlier and then outcompetes the other neurons, and so you get the sparse.
Right. That's
activity.
I'm not talking about that. That's the basics of this. I'm just saying right here, it's the first time I've heard this. This is nothing. This is the layer 5 cells. This is this bursting versus regular spiking. You remember also in our paper we just wrote, we made the analogy between that and the bursting and regular spiking in the relay cells. So in the thalamus, the relay cells have these two modes, burst and spiking, and so do the layer 5A cells. And we said maybe that's related to their ability. That's someone has to do with their ability to do the orientation transform, because we expected both those cells to perform orientation transform. Right, we're getting really down in the weeds here, but this is an interesting paper, but I'm not sure it's.
Yeah, but yeah, I think you're right that these are the motor output cells, in
Right.
this case A5P, because here they say also that they are the sole cortical output beyond the midbrain.
Right.
And yeah, so I think that's those are the motor output cells. Yeah, let me see if there were other things that I wanted to mention real quick. I got that. Let's see. Oh, this one is not matrix cells, but it was a fun paper. You're probably aware of it anyways, but maybe just to frame the importance of learning sequences all across the cortex, not just for learning melodies. This is the paper, I think they say, this is the first one that shows that sequences are also learned in V1 and that it's pretty much directly modeled within V1, not just some higher order region that models the sequence and then tells V1 what to expect.
What year was this paper?
2014.
Well, wasn't this your supervisor, Scott?
Yep, that's my old PI.
Wow. Was this before or after our neuron paper came out? I don't remember. I wonder if they cited us in that one.
Viviane is 2016. He's definitely familiar with Numenta stuff, but yeah, I don't know if this was.
That was 2016, so it was
Oh,
before.
this was before our paper. Interesting.
Yeah, so
I remember he came and visited Numenta. I think it was during
I think so.
my
Now it's
first
coming back
year
to me.
of my PhD.
Right. Now it's coming back to me. But at the time we wrote the paper, we didn't know about this.
So yeah, basically they have a pretty simple setup. They have rats and they show them sinusoidal gratings at different orientations in a sequence. And so the trained rats for 4 days, they see A, B, C, D. And then on day 5, they're tested on the same sequence and a new sequence and the same sequence, but twice as slow. So instead of 150 millisecond interval, it's a 300 millisecond interval. And then there's a control group that's just not being trained. And so, yeah, you basically see that there is a significant sequence effect for the learned sequence compared to the sequence that's new and wasn't seen before. But then also it
So as
doesn't
in,
seem
sorry,
like
you have higher activity or lower activity if you can predict it?
higher activity, like higher spikes here, and then also the kind of histogram down here is higher.
Yeah, when it is correctly predicted.
Yeah.
Okay.
So the important caveat here is that this isn't spikes. This is dendritic currents, really, because this is local field potentials. So we don't know whether or not that really translates to spikes. We just know there's a lot of dendritic activity. So it could be, a lot of inhibition or whatever, and this
Or
is
it could
layer
be,
4.
it could be the dendritic spikes anticipating the next input,
Yeah.
So yeah, there's definitely some kind of learning effect of that sequence. And then they
That
also
also
tested-
generalizes. Oh, sorry. Just to check my understanding. Yeah, and that generalizes to the slower sequence as well.
Not too well. So they actually argue
Oh, they
that
argue
it's
that that's the same.
not really generalizing. I think,
Okay.
I mean, yeah, I didn't have enough time to look at it in detail. To me, it still seemed like it was better than it would have been for a completely new sequence.
Right.
But I'm not sure. I don't
Yeah,
want to
visually
claim
it does
that.
look different.
But yeah, they did. I think this one shows it. No, maybe it's not that. Yeah, maybe I stay with this one. But I mean, it's like, as far as I understood, they mostly look at the first next event. So maybe it was just too short of a sequence for it to recognize that it's half the speed and to adjust it and then make the new correct predictions.
It also may be that animal can't make a half a speed, it can't make it go half a speed. Or it could, and we're doing this in V1. I mean, there's so many unknowns in experiments like this. I think what you can only walk away with a certain amount of conclusions. just this, as Scott just said, oh no, we're looking at dendritic currents. Oh, we're local field potential. Well, that's all kinds of crap could be happening there. Local field potentials also include, as I think Scott mentioned, inhibitory cells firing, and oh my gosh, what the hell's going on?
Yeah, I think the main nice takeaway I had from this was that, yeah, sequence learning, I mean, that we were already kind of, it was already clear to us, but that it's happening everywhere, even early visual cortex. It's not just some higher order region that models sequences, or special regions that are for temporal models. Yeah. They say here, like, in this framework, visual cortex is visual not because it's specially suited to deconstruct the visual scene, but because it receives input from the eyes. Basically, yeah.
One time, not Scott, a researcher who studies rats told me, he goes, or monkeys, he says, we train these animals for days, sometimes weeks, on these simple stimulus over and over and over again. It's hard to imagine what the animal's thinking. They might be going crazy. It's like being locked in solitary or something. It's like, they're just like, stop it. It's like, we really have no, the animal's not thinking, like, oh, an interesting pattern. It's more like, oh my God, I'm being drilled into my head, So it's another interesting thing to think about. These are not natural. I mean, I like the conclusion of this paper, but these are very, very far from any kind of natural world stuff that's going on out there.
Right. Yeah. Maybe last small thing to mention is they also tested omitting an element, both in like starting the sequence with A, like it was learned, or starting the sequence with a different letter that was not learned, and then show that like it seems to make a prediction here, but not that has a different response with the new element.
Yeah, that's pretty definitive.
Yeah.
Yeah, nice. That's a cool paper.
Okay, I think those were the main parts. I'm sorry, I wanted to make a better presentation, but
that was good. Will you share the consciousness paper?
Yeah, I can share the papers.
Yeah, the consciousness paper.
Sure.
It might be easier to read, fun to read. What did you put in Slack, or you?
Oh, I can share it after this.
I don't know. Did you just share it? I don't know where you shared it to.
Not yet. No, I'll put it in Slack.
All right. Whatever.
Okay, that was all I had. I don't know.
So do you think you have enough to get started on this?
Yeah, I have to sit down on the time component again and think about it, because with Monty we also have currently we have discrete time steps. So we're currently not really working in like a continuous time environment. The environment is static, and then the steps are just as Monty moves, it sends a new observation. So I think there might be a couple of tricky parts to figure out as well, like when does Monty sample observations from the environment and so on. And how do we take into account that? Yeah.
That's going to be a hard thing to change. That's going to take some pretty big engineering thinking to get rid of discrete time steps.
Yeah, and we're going to need to adjust the simulator, the environment, to get objects moving around as well, according to some whatever rules we set up.
Yeah, what are they called? Is it event-driven cameras, where basically as soon as something changes, then they send information? So I think that's a bit closer to the retina. That basically.
But just right now, I assume Monty takes its time, right? It gives a new time step, it processes everything, and it gets the next time step, right?
Right. So
fundamentally
with the
not
sensor
real time.
module, with the sensor module, we could just stream data from the environment at a high frame rate, and then whenever it detects a change, it sends it to a learning module. But then, the problem is the learning module might take some time to process it, and in that time, other things might have already been happening in the environment, and that doesn't get stepped until the learning module is done. So, I think there will be some tricky things to.
Well, I think I mean, even humans, right? You know, we have a certain rate at which we can process information. If things happen quicker than that, we can't perceive it, or we get lost. And so it might be that you could simulate real time, but run slower. You know what I'm saying? Like, okay, you know, if a human can't process anything less than 300 milliseconds, for example, you could just let Monty take as much time as it needs before you go on to the next, quote, real-time input. I mean, it doesn't have to be real time. It has to just be simulate real time.
Yeah, because we've been talking about. Oh, I was just gonna say, yeah, with the CMP, we've been talking about how we could make it more kind of asynchronous and sort of support some of this stuff. And it feels like it's doable, but as you say, Jeff, it's it's a lot of big questions and it's a lot of engineering to like get it set up so that for the moment we might ignore that problem and just assume we have kind of a global clock, global ticker.
All right.
Um,
We'll have to deal with it sometime.
For first prototype, we can just have an environment or data set that's simplistic enough that we can step time in the environment.
Right, right. I think that's fine as long as when you actually, if you rely on this sort of real time, you know, real world clock, at some point you might have to get rid of it, and then you might have a lot to undo. So just be conscious of that. I don't. This is a problem that's been dealt with in computers for ages. So we're not the first people thinking about it.
So yeah, I think the main tricky parts are around the time component right now, but otherwise, I think we could do
if there's anything that's tricky that you can phrase as a problem, like what's the solution to this problem, you post that.
Yeah, I think in terms of like theory, it's like, how do we know when a sequence repeats, and how do we invent events in a continuous behavior?